Today we're going to follow the incredible journey of a tiny piece of tissue, a biopsy sample, as it travels from a doctor's office into the hidden world of a laboratory. It's one of the most important processes in all of medicine, yet most of us have no idea how it all works.
You know, it's a procedure that millions of people go through. But what actually happens after the sample is taken? For most of us, it's a total black box. Where does that little piece of tissue go, and how on earth does it turn into the answer that you or a loved one might be waiting for?
Well, it embarks on this complex, almost secret journey right into the heart of a lab. And in this explainer, we're going to pull back the curtain on that process, which is without a doubt the absolute gold standard for diagnosing disease. So, the science that makes all of this possible? It's called histopathology. And if you break that word down, it's pretty simple. It's the science of seeing disease right inside our tissue. And to really get how important this is, just think about this for a second. Blood tests can give you clues, right? And imaging, like an X-ray or MRI, gives you the big picture. But histopathology? That gives you direct, undeniable proof straight from the source of the problem. So what is a pathologist actually doing? Well, they're basically detectives at a microscopic level. They're studying diseased tissue, looking for those telltale signs of how a disease messes with the very structure and architecture of our cells. Okay, so take a look at this. This gives you a really clear idea of what a pathologist is searching for. On the left, you've got healthy tissue. See how the cells are all uniform and orderly? Everything's in its place. But then you look at the right, and it's just chaos. This is what pathologists call pleomorphism, which really just means many shapes. The cells are all different sizes, the nucleus is dark and way too big, the whole structure has just broken down. All right, the journey begins the very second that sample is taken. This first stage, just getting the tissue safely from the patient to the lab, is called the pre-analytical phase. And you might be surprised to hear that it's one of the most critical parts of the entire process. So why is it so critical? Because believe it or not, this is where most of the mistakes can happen. A simple delay, a wrong label, or even just letting the tissue dry out. Any one of those things can lead to a completely wrong diagnosis. The stakes are just sky high, right from the get-go. The very first, and you could argue the most crucial step, is something called fixation. This is all about preserving the tissue. It's like hitting a pause button, freezing it in time to stop any decay and keep its structure exactly the way it was when it was inside the body. Now, fixation has two main jobs. First, it stops autolysis. You can think of that as preventing the cells from self-destructing with their own enzymes. And second, it stops putrefaction, which is just destruction caused by bacteria from the outside. So it stops threats from both the inside and the outside. And this whole process, it is all about precision. You have to get it just right. For instance, the tissue can't be thicker than 5 millimeters. Otherwise, the fixative can't get all the way to the center. You need at least 10 times the tissue's volume in fixative for it to work right. And the timing is super critical, somewhere between 6 and 24 hours. Too little, and the tissue decays. Too much, and it gets all brittle. Okay, so once the tissue is perfectly preserved, it's time for a complete transformation. To see it under a microscope, this soft, wet sample has to become a solid block of wax, firm enough to be sliced thinner than a piece of paper.
But this creates a really fascinating problem, right? Our tissue is about 80% water. The goal is to get it embedded in paraffin wax. But as we all know, water and oil, or in this case, wax, they just don't mix. So how do you solve that puzzle?
Well, the solution is this really clever three-step replacement game. First up is dehydration. You use alcohol to gradually pull all the water out. Step 2 is clearing. A solvent, usually something called xylene, is used to replace the alcohol. And finally, infiltration. Now that the xylene's in there, you can finally replace it with hot, molten wax, which seeps into every last nook and cranny of the tissue. Now, as the tissue is put into that final wax block, maybe the most critical manual step of the whole process happens: orientation. I mean, just think about it. If you have a skin sample and it's placed on its side instead of flat, the pathologist could completely miss the specific layer of skin where the disease is. The diagnosis could be literally hidden in plain sight just because of how it was positioned. So now our sample is a solid block of wax. The next step is to get it ready for the microscope, which means slicing it with incredible precision and adding some color to reveal its secrets. Using a super precise machine called a microtome, a technician slices that wax block into sections that are just 4 micrometers thick. Now, to put that into perspective, a single red blood cell is about 7 micrometers thick. So we're talking about a slice that is thinner than a single blood cell. That is an absolutely incredible feat of precision.
But here's the thing. Even when it's that thin, the slice is pretty much transparent under a microscope. It needs to be stained to see anything. The workhorse stain is called H&E, and the chemistry behind it is actually surprisingly simple. Opposites attract. You've got hematoxylin, a basic, blue dye that's attracted to acidic parts of the cell, like the nucleus. Then you've got eosin, an acidic, pink dye that's attracted to the basic parts, like all the cytoplasm surrounding the nucleus.
And just like that, the slide is ready. The tissue's long and complicated journey is nearly complete. All that's left is for a pathologist to look through a microscope and read the story it has to tell.
The final result of all this work is the histopathology report. It includes the gross examination, which is just what the tissue looked like to the naked eye, and then the really detailed microscopic findings. From all of that information, the pathologist makes a final diagnosis. And for cancers, they'll also determine the grade, basically how aggressive the cells look, and the stage, which is how far the disease has spread.
But sometimes that standard H&E stain isn't quite enough. Pathologists have a whole toolkit of special stains they can pull out. If they think they might be looking at a fungal infection, they'll use a PAS stain. If they're looking for signs of tuberculosis, they'll use a ZN stain. Each one is like a unique chemical key, designed to light up a very specific clue inside the tissue. And of course, the technology here is always advancing. Modern techniques like immunohistochemistry or IHC are amazing. They use antibodies like tiny programmable tags to light up very specific proteins on cancer cells. This doesn't just confirm a diagnosis, it can actually tell doctors which targeted therapies are most likely to work for that specific person. And so the tissue's journey, from a simple biopsy to a complex, beautifully stained slide, is really a hidden marvel of science and precision. It's a whole process designed to give us definitive answers. But as this technical journey ends, that final report marks the beginning of another, deeply personal journey for the patient.